Welcome everyone. Today we are going to learn about lipid anchored proteins. In my previous video, I have discussed about that there are three major categories of proteins present in the plasma membrane. One is called integral membrane protein, another is lipid anchored protein, and third is peripheral proteins. Now today we are going to learn about lipid anchored protein. Lipid anchored proteins attach with the plasma membrane with the help of lipid molecules. So there are three major types of lipid anchored proteins present. One is called acylations, where fatty acids are actually attached with the proteins, and then is prenylations and GPI anchored proteins. I will talk about all these three type of lipid anchored proteins. At first, I will talk about the acylations. Now, in case of acylations, the proteins are actually cytoplasmic proteins because in case of plasma membrane, there is two surface. One is exoplasmic, that is the exterior of the membrane, and it is cytosolic, that is present in the cytosols. So as you can see, there is two lipid bilayer. So this type so proteins can be present from outside of the membrane or inside or cytosolic. So here in case of acylation, cytosolic proteins are actually attached with the help of fatty acids. Second is the fatty acid chains are actually myristate or myristic acid or palmitic acid. Okay. Third point is the protein has actually N-terminal glycine molecules. You see here, there is a glycine molecules present. Those proteins contain N-terminal glycines. These proteins can make amide linkage with the fatty acids like myristic acid or palmitic acid. So you can see here that amide linkage between terminal amino group and myristic acid. So myristoyl anchor it could be. So acylation could be myristoyl anchor or palmitoyl anchor. If the fatty acid is myristic acid, so we call myristoyl anchor. If the fatty acid is palmitic acid, so we call palmitoyl anchor. In case of palmitic acids, thioester linkage actually occurs because here cysteine amino acids and the palmitic acid actually makes the bond. But in case of myristic amino acids, glycine actually makes the bond. So it's a amide linkage, and in this case, thioester linkage occurs. Now, these modifications or acylations actually occurs during the synthesis of the protein on a ribosome. Now let's talk about one example of acylation, that is V sharp. So we call SARC proteins. Basically, this is a mutant form of cellular tyrosine kinase. So this is actually an enzyme. Tyrosine kinase enzyme means this enzyme can phosphorylate tyrosine amino acids of other proteins. Okay, so SARC family members are actually tyrosine kinase, but when these tyrosine kinase actually are mutated, we call a V shark. Basically, they actually induces abnormal cellular growth and causes cancer. But this protein can does this so only when it is myristoylated at N-terminal. So this acylation is very important for the functioning of V-sharp protein. All members of shark family are myristoylated. Also, other family members of the shark family are actually myristoylated. So the V-shark protein also. Now membrane attachment through a single lipid anchor is not very well. So as I have mentioned, by acylations, myristic acid can be attached or palmitic acid can be attached. So by a single lipid anchor, it is not very strong. With the attachment is not strong. So what happens for most shark kinases? The second lipid modification is palmitoylations. That means the attachment with the palmitic acid. So the first attachment is a myristic acid attachment, that means fatty acid like myristic acid attached with the protein, and the second attachment is with the palmitic acid. So these modifications, now the second modifications actually occurs in response to an extracellular signals. So when extracellular signals come, this type of modification occurs and a strong attachment of the protein occurs with the plasma membrane. Now when the signaling pathway is turned off, the palmitic acid, that is the second lipid modification, is actually removed, allowing the kinase to return to the cytosol. So it is actually turned off, the signal is turned off by this way. Now let's talk about prenylations. In case of prenylations, as you can see, it is also the cytosolic proteins that are attached. So this is the cytosolic phase of the protein. So cytosolic protein attached with prenylations. Now in case of acylations, fatty acyl chains are attached, so it is called acylations. But in case of prenylations, hydrocarbon chain of 5 carbon isoprene units are actually attached with this protein. Now 15 carbon farnesyl or 20 carbon geranyl not, it is not a fatty acid but hydrocarbon chain are actually attached with the proteins. So this 5 carbon isoprene is the unit. So when it makes three bonds or five carbon attaches, it form a farnesyl group. When 10 carbon actually attached with, we call it geranyl, and then another 10 when actually add, we call geranyl because then it contains 20 carbon. So geranyl means 10 carbon hydrocarbon. When 20 hydrocarbon attached, then called geranyl, and in case of 15 carbon is called farnesyl. So here is hydrocarbon chain present, not, it is a not a fatty acid chain. So it's different kinds of actually hydrocarbon attached with the proteins. So it's called prenylations. Now here, at or near C terminus of the protein, a cysteine amino acids actually makes the bond with the hydrocarbon chain, as with the palmitic acids, you know. So in case of palmitic acid, thyester bond form. In case of prenylations, thyester linkage occurs between cysteine and prenyl group. Now this C terminus cysteine is actually a group of amino acids called CAAX box, where C means cysteine, ALA means alanine, and X means any amino acid. So there is a particular four codes of amino acids present called CAX box in the C terminus, where the cysteine actually makes the bonds with the prenyl group with the thyester bonds, and this alanine, and X is actually cleaved. Thiostyle linkage between cysteine and prenyl of hydrocarbon occurs. This modification occurs post-translationally, as I have mentioned in case of acylation. That is simultaneous the production of the protein. This modification occurs, but in this case, it occurs post-translationally after the complete generation of the protein. In some cases, here also, a second geranyl or palmitic acid is attached or linked to a nearby cysteine amino acid. So the second fatty acids attachment or isoprene units are actually attached with these proteins to make the attachment strong. Examples of prenylation protein is RAS protein. RAS protein helps in cell signaling, and RAP protein actually present in the intracellular vesicles that helps the vesicles to attach with this target membrane. Now another type of lipid anchor protein is GPI anchored protein. GPI means glycosyl phosphatidylinositol anchor protein. Now I have already mentioned that in case of phosphoglycerides of lipid molecules in the plasma membrane, one species of phosphoglyceride is phosphatidylinositol. What is phosphatidylinositol? Here two fatty acids is attached with a glycerol with a phosphate and an inositol. As you can see, this green is actually inositol. Inositol is actually a sugar molecules that attached with these fatty acids, and actually is a phosphoglyceride. So backbone is a glycerol. There is two carbon attached with the fatty acid chains and a phosphate attached with the inositol. So basically the basic structure of GPI is actually phosphatidylinositol. Now with the phosphatidylinositol, there is glucosamine sugar molecules, mannose sugar molecules, and phosphoethanolamine are also present. So this is the whole structure of GPI. Now protein looks, this is the precursor protein. It is present in the endoplasmic reticulum lumen. Now in my previous video I have mentioned topological equivalence. That means the lumen of any intracellular organelles is actually similar with the exterior of the plasma membrane. If you have not watched this video, you can watch the topological equivalence video of my plasma membrane, so you will get a clear concept of what I am going to talk about. Basically this protein actually faces the ear lumen, and the modification occurs in the ear lumen. So there is a protein called GPI transamidate that cleave the protein here and attach with the preformed GPI anchor, and it looks like this. Now this protein actually move from endoplasmic reticulum to the plasma membrane. Then this protein actually not present in the cytosol. They actually face the exterior of the cell. So this is called topological equivalence. The lumen of intracellular organelles are actually topologically equivalent with the outside of the cell, because this is the cytosol, and cytosolic proteins actually face the cytosol. It's all actually always it occurs. Now, in case of GPI anchored protein, the proteins are actually face the exoplasmic domain of the lipid bilayer, so it's exoplasmic proteins, but actually modification occurs in the EL domain. Now, phosphatidylinositol has two fatty acid chains. By these two fatty acid chains, basically they attach with the lipid bilayer. This is for other phosphoglycerides. Okay, this is simple. Now, C terminus of proteins attached with phosphoethanolamine. So this portion is actually phosphoethanolamine, and here the C terminus of the protein, because this is the N terminus of the protein and this is the C terminus of the protein. C terminus attached with this phosphoethanolamine. This modification occurs post translation. As you can see, the protein is already made. So in case of GPI anchored protein also, the modification occurs after the translation of the protein. Examples like Thai-1, placental alkaline phosphatase, acetylcholine esterase, and cellular prion proteins are example of GPI anchored protein. So this is all about the lipid anchored protein.